So last time I talked about some of the basic organelles of a cell. Remember, organ is an organ. L is little, so the little organs of the cell. And so I made the cell again here. You can actually see the phospholipid bilayer, the cytoskeleton, the Golgi, the endoplasmic reticulum, both the smooth and the rough. You can see the nucleus here and the DNA, which I just put in there. And I put a couple free ribosomes here. But I didn't talk about one of the other organelles that's actually really important for cells, and that is the mitochondria. Now, the funny part about the mitochondria is a lot of people tell you it makes the energy for the cell. The truth is, in human cells, the energy is actually taken by diet, and it's then ended up whatever the sugars usually we eat are actually broken down into certain components. So if you think about it, we made our DNA into mRNA. It left the nucleus and went to ribosomes, and it made certain enzymes, which are protein catalysts. And what they do is they take some of these will take sugar, and I made one round one here, and it will break it. And when anything is broken, it releases energy. Whenever you build something up, if you want to make something, you have to add energy. So if that circle I made was broken in a 2, you release energy. And that energy you release, we'll have 2 now, these sugars here, that energy that gets released by the break ends up being used to make other things that you can use to store energy and use it in the cell later. I will talk a lot more about that later. Now, that gives you a little bit of energy. For every sugar molecule, which is what most cells are going to use initially for energy, it gives you roughly 2 ATP when you break it here, and you can continue breaking it. Now, this doesn't use anything other than breaking. Now, we do have another organelle, which I'm going to move my sugar out of the way, called a mitochondria. And I made it here. It does have 2 membranes, and I did make it red because most textbooks have it in red. Well, the sugar will actually be sent to the mitochondria, and in the mitochondria, it will be broken down more and more until you end up getting to pretty much carbon dioxide and water. And that's actually going to leave. And so you need sugar to come in, and those are the protein structures that I actually didn't draw this time, but they would be on the cell membrane to help you pretty much get stuff in, receptors, a lot of different things. So now we actually have the basic components of the cell. I still have the vacuoles, but the reason I actually did the first area first was because those are the organelles that we're really going to talk about most when we deal with how you make bone. And before we actually know how to make bone, I actually do have to talk about basic embryology. And so I'm going to have another cells, and I'm going to show you what I'm doing. Now, at this point, the different colors are not going to represent any specific structure of a cell, but rather just to be able to show you how cells work. So our big cell would actually be this here. And this giant cell would go into a rapid division and start forming pretty much a lot of little cells and make a big ball of cells, little bit that look like this, which would be called a morula. And you have thousands. Now, a lot of times people ask about twins. Usually when there are twins, this is what happens. One of these goes on and starts making another morula of itself, and so you can end up having 2 different types of twins. And there are different types of twins, which I'll mention later. Now, this actually ends up becoming a big ball of cells, a hollow cell, and it ends up becoming a layer outside, and this thing called the inner cell mass. I made it yellow so you could see it, but the inner cell mass is going to do something really cool, which I'm going to show you with 2 different colors. So by week 2, what's going to happen is the inner cell mass is actually going to become 2 different types of cells. One of them, which I'm going to use the blue as the epiblast. Epi means top, blast means maker. The bottom cells are going to be made into what's called a hypoblast. Now, if you notice, there's an area between them where they touch. And if we were to cut this, what we would see is we'd see these 2 layers, and it would form this area here. The epiblast is the top layer, the hypoblast is the bottom layer. Now, underneath and here, inside the yellow tube, which is our hypoblast, you have your yolk sac. Above it, in this blue tube I made, you would have your amnion. And so these would actually be cut, and you can see these here, so you can see how they would look. But what would happen is these 2 would be 2 discs. So if you actually looked at it from the top, you just see a flat layer of cells. As you have two cell layers, we call it a bilaminar disc. Bi means two, laminar layered, and so it's a bilaminar disc, two-layer disc. This area here is the only thing that's going to become human. The rest of it are cells that are going to be doing different things, which I'll show you how what happens later. And things are going to change. There's actually going to be some growth, and cells from the epiblast are actually going to migrate between the epiblast and hypoblast and make a third layer. I'm going to make this one red for now so you can see what it is, because in about the third week of pregnancy you have 3 cell layers. And as you have 3 cell layers, now we call it the ectoderm, the mesoderm, and the endoderm. This area here will become your gut tube, most of your internal organs, and the outer layer, as well as other things, which I'll show you. So we're only interested in these 3 layers here. So what you're noticing is most of this we can actually not even look at. Yes, you do have a yolk sac, and it is used for making primitive blood cells sometimes. But for me to actually be able to show you that, I'm going to actually just see how things happen. I'm just going to look at these 3 layers. Now remember, this is week 3 that this is happening. So you're talking about the beginning of the month, the end of the month. So what I did is I removed the circle so you can just see the trilaminar disc, 3-layer disc. The outermost layer here is going to be called your ectoderm. The red middle layer is your mesoderm, and the inner layer is your endoderm. And what's going to happen is the ectoderm and mesoderm is going to grow a lot faster than the endoderm. So it's going to rotate around. It's actually going to make itself into almost a donut. So you're going to have the mesoderm on the outside, the endoderm on the inside, and the ectoderm on the outside. And you will actually connect in the front here. I didn't use enough clay to do it, but you'll actually have the 3 cell layers. The innermost layer will become your gut tube. The outermost right now is just your outer layer, and the mesoderm is what's going to be the majority of it. And again, this is pretty much the week 3, early week 4. So you're talking about still the first month is when all this is happening. So far, all these cells can actually break apart and make different types of cells. And so that's actually what we're actually showing, is you have this circulation in it, and you can see it in the human body. Now, as we do now have a specific symmetry, we actually can see how your arms will actually come out from the side and legs will come out from the side, which I'll show you later. And what's going to happen is you're going to have a back part, or technically a top part here, which is dorsal. Over here is ventral. Now in humans, our dorsal, when we go in embryology, becomes our top part. So it ends up becoming our posterior side. Posterior means back, anterior means front. Lateral would be to the sides. These are actually words that will help you in anatomy, and I'll mention them again later. But you can see how the circle ends up becoming. And I'm actually only going to talk about the circle right now, because what I'm going to do is I'm going to talk about the axial skeletal system first, once I talk about how everything is made. And so with that, I'm going to modify a little. I'm actually not going to put the mesoderm. What I'm going to do is anything that's mesoderm in origin, I'll make it red. The reason is you're going to have parts moving. And so give me 3 seconds. So here you actually have the ectoderm, the endoderm, and the mesoderm is actually going to be everything inside. But you're going to start forming different things. One of the things you're going to form is a structure. This is about the fourth week. You end up forming something called the notochord, which is here. And it will cause a pulling of the ectoderm. And so what you're going to have is the ectoderm is going to fold, leading to a neural fold. And eventually, it's going to be pulled in to the developing organism. So I'll show you those 2 things. So what happened is part of the ectoderm here got pulled in. So you have a neural fold that I showed you. Then you end up forming the neural tube, and you have 2 groups of cells on the outside called the neural crest cells. Now we're going to be dealing with bones mainly, but I just want to show you how certain things form. The notochord is going to be important for the axial skeletal system. The neural tube is going to be eventually going to be something that resembles a brain, and the neural crest cells will actually develop even more. And so what I'm actually going to do is I'm going to make this would be a model that would have been cut if the developing structure would be this ball here, it would have been cut across the side here. Now I actually want to do one where if I were to turn this around, because you can actually see certain things develop and a little bit later. But what is going to happen is you're going to have another group of cells condense first in the mesoderm here, and this shouldn't move, and on the other side here, called the somites, and the somites are going to divide into three parts. One of them is going to be called the sclerotome. Sclero means hard, and so tome means layer, so the sclerotome will actually make a membrane around the neural tube, which I'll show you a different view of this so you can see what I'm talking about. So at this point, I'm actually showing you the outermost layer here is the ectoderm. The mesoderm is that red thing here. Actually, it would be anything between the neural tube and the ectoderm. The notochord here, and the gut tube here. Now what we're really going to be interested in when we're dealing with bones, in particular of the axial system, is the neural tube and the notochord. And what happened, and the somites that I told you about, forms a myotome. And so the myotome actually just really forms a layer over the neural tube. While that's happening, the neural tube is doing some other interesting things. So pretty much what starts happening, and I'm going to pull the neural tube out so I can do certain things, is the top part of the head, the top part of the neural tube, the anterior portion, starts ballooning, giving you a head. That means on this guy, this would actually narrow a bit, and this would become bigger. So now you have a head end called the cranial end, and a tail end, the caudal end. And if you notice, we're starting to look like something out of a fish. As that happens, like I said, you have the sclerotome going around it, and it's going to lay down a membrane called a mesenchymal membrane, which is going to go on almost all the neural tube. And the notochord will actually be important again, so I'll talk to you about that later. But now I actually want to show you what's happening in the sclerotome, so you can see how bone is being made, and then I'll show you about which bones are made like that. So what happens is, in the mesenchymal membrane, some of the cells specialize and form blood vessels like arteries and veins. You do have some nerve innervations that actually go through there, and cells specialize around that area, which are going to be called osteoblast. Osteo means bone, blast means maker. And each one of these cells, which are made white, will start secreting protein matrix to help make the basic structure of bone. But it's not going to be alone. The other thing it's going to do is it's going to take the calcium and phosphate from the blood vessels and bind it to that. And so the cells here are going to be trapped in. The way they make the protein is just like we said at the beginning. You have the DNA for the protein. It gets copied to RNA. The RNA gets copied into the RNA copied into protein. Usually as this is secreted, it's going to be made in the rough endoplasmic reticulum. And through exocytosis, it goes from the rough endoplasmic reticulum to the Golgi, and then gets secreted out. So all the area between these white cells that I made are going to end up having a bunch of proteins going in there. So even though I'm making it, I don't want to use the red too many times. And so you end up having proteins form all the way around these cells. And to make sure that the cells are not trapped without the nutrients it needs, the cells actually start forming these little projections, which are going to try to go towards a blood vessel. And they're going to make a membrane around there. And so you're going to have proteins and calcium phosphates around here, and it's going to make a structure, and the cells are going to be trapped inside. So I'm going to show you how this would look in bone tissue. So in the bone tissue, compact bone in particular, you'd have these circular structures which were formed by the osteoblast. You'd have little holes here. You can see here, here, and here, which are where the osteocytes here would be. And you'd see these little lines going from the osteocytes to pretty much the central hole, which is called the central canal. And you would have little areas of cells pushing itself in there. As these are little canals, they call canaliculi. And you wouldn't just have one of these structures, which is called an osteon, but you'd have tons of them, making this go around. You'll notice that this looks like a tree. And so this can actually take a lot of force on the axial, but on the side, it can take forces, but not as much as axial, and that's actually going to be really important. Now you will notice that I left a little hole here, because while you do have these circles called concentric lamella, You also have interstitial lamella, which are pretty much the same type of structure inside here, filling in the gaps. And so that's actually compact bone. Spongy bone would be made as well, which the reason it's called spongy is because the fibers, I use this, are kind of going everywhere. You do have cells, you do have all the other stuff. And so you can have compact bone and spongy bone just like that. And over here, so a lot of times you'd have a cavity. We don't need those. So you have the outside. It is usually covered by a membrane called the periosteum. You have the compact bone, spongy bone, and then you have the medullary cavity if you have one past it. So you have the outer layer, which is cortex, inner layer, which is medulla, by the way. And so now you actually have the basic structure of pretty much the bones that are going to be in the axial skeleton system. There is another tissue types that I would want you to know before we actually talk about bones, so you can get a feel for what we're looking at, and that is the cartilage. And cartilage, you'll notice that with bones, I showed you that it has blood vessels. Cartilage doesn't have the same blood vessels. In many ways, if you actually were to make cartilage, the most basic cartilage would be hyaline cartilage, which pretty much would be the matrix, a big hole, a couple of holes where cells would go. And if you remember in bones, the holes in bones here were called the would be called the lacunae, and the holes here would be lacunae as well, and you'd have a little cell inside each one of these holes, and those cells in cartilage will be called chondrocytes. Chondro meaning cartilage, cyte meaning cell. When they are making it, they're called chondroblasts. And you do have three different types. You have fibroelastic and hyaline. Hyaline is the most common, and I will mention a little bit about those two later on. I hope you enjoyed this, and I intend to go into the other tissues of the bones later. Goodbye.